Today our speaker is Rob Skelton from the Fynbos node. He's also affiliated with UCT. Are you affiliated with UCT, Rob, or you just hang out there a bit?
Yeah, I just supervise a bunch of students.
Okay, so he's actually, but he belongs entirely to SAEON. So Rob is a FLAIR fellow, which is a Royal Society funding program for exceptional young scientists, and he's also a P-rated NRF researcher, which means that the NRF think that he will be a future leader in his field. Rob, I've known Rob for quite a while. We did our undergrad together. I was very surprised to see how Rob has become such an influential academic. In undergrad, I thought all he was going to do was play frisbee for the rest of his life. So those of you who are trying to forge careers in science, there's hope. Even a lowly frisbee obsessed player can become a influential scientist. So Rob's a great example of that. Rob's research is focused around plant ecophysiology and how plants cope with and how they're adapted to drought. And yeah, I've been starting to collaborate with Rob, and his research is really interesting, and I'm really looking forward to finding out all about it. So Rob, take it away.
Thanks, Glenn. It's a very generous introduction. The frisbee is instrumental to my research, so those two things, you can't separate them. All right, so hopefully I'm going to share my screen. Can everyone see this? All good? Okay, great. Great, so as Glenn mentioned, I work on plant physiology, and what I would like to present to you today is how we can use plant physiology, some of the principles of plant physiology, to understand the impacts and extent of global change on natural vegetation types. So a little bit more background, a little bit more context for my research. I started off as a SAEON PhD student. I received a SAEON bursary, and I researched the physiology of mountain Fynbos at Johannesburg. I then received an NRF postdoctoral abroad scholarship, which allowed me to travel to Tasmania in Australia, where I conducted research in some remarkable ecosystems there. I then crossed the divide. I went north to California, Berkeley, where I took up a position as a research, a postdoctoral researcher at the University of California, Berkeley, working on the physiology of oaks. And so now I've come full circle to return to the beloved mountain Fynbos, the vegetation type of Southern Africa. So today I'll be leaning on two case studies to showcase the ability of physiology to address some of these long-standing challenges, long-standing problems in physiology. So one of the questions you might be asking at this point is why plants, why plant physiology? I might be preaching to the choir for some of you, but plants underlie many of these larger processes that occur in the world. So plants, through the process of evapotranspiration, return vast quantities of water to the atmosphere, which then drives local climate and global climate. Plants also provide they often filter water in watersheds. So here's an example of where there are two rivers here that start in this site, but one of them is particularly important, the Sonarent River, which then flows into the Kloof Dam, which supplies drinking water for millions of people. So plants are incredibly important in influencing runoff, influencing the filtering of water supplies. So they're incredibly important for these these larger processes. In addition, in the agricultural setting, plants are the basis of most of our food products. So we should be particularly interested in how the function of plants is affected by things like abiotic stress, drought, and fire, and other elements of global change. In addition, plants are the basis of natural resources like timber. It's not so prevalent in this photo, but we get a lot of natural products from natural vegetation. So we drink tea from natural products, we use timber, etc., etc. The benefits of natural vegetation and healthy plant functioning is almost infinite. The last point is that plants provide the basis of much ecotourism, even in the savannas and areas where people go to watch the animals. Plants are the basis of the food chains in those areas. So happy, healthy, functioning plants make people a lot of dollars. They bring in a lot of money through tourism. Of course, we're all familiar with the idea that humans are changing the atmosphere, that we're changing the earth, we're transforming the earth. One of the ways we're doing that is we're producing carbon dioxide in extraordinary amounts, and the predictions based on some of the models are that if we continue as normal, we might return to conditions last seen 50 million years ago in the early Eocene, when I think forests were flourishing in Antarctica, so the world was very, very different. Even the best case scenario suggests that there is likely to be some change to the temperature and associated climate. So really the question that we should be asking is how are natural vegetation areas, how are plants likely to be responding to these changing conditions? In Sub-Saharan Africa, and in particular the southern tip of Africa where we are based, water is in incredibly short supply. So we live and function in arid and semi-arid and even hyper-arid systems in the deserts, the Namib Desert, for example. And the impacts of global change are likely to be, at least pertaining to water, are likely to be particularly acute in these semi-arid to arid areas. One such example is changes in rainfall are likely to non-linearly affect changes in surface water supply, making arid areas even more arid, so disproportionately affecting runoff in these arid areas. And already we're seeing the impacts of such global change on the natural vegetation. So we're seeing mortality of here's Allodendron dichotoma, the quiver trees, and then a handful of other examples of widespread mortality events of plants. I think this is from the Sahel region. And so the question is, what is the likely impact of these global change events on the natural vegetation of the region? In addition to the dieback caused by drought events and other global change drivers, there are many drought events that don't cause extensive dieback, that cause loss of function in these systems that are so incredibly important. And this forms the basis of my research, which is to examine the extent and impact of natural change on these global change on these natural systems. So before I dive into two case studies, I'd just like to provide a brief overview of some of the fundamental terminology that I'll be leaning on, some of the terminology that's become jargon in the world of plant physiology. So we talk about water potential. Water potential is just the water status of the plant, and to understand why water potential is important, we need to understand how water moves through a plant. One of the things that makes plants that much more interesting to research than animals, of course, is the idea that plants don't have a heart, so they can't pump water to the tops of their canopies, the top leaves. Water is passively drawn up through the process of transpiration, where the sun heats up the mesophyll tissue, heats up the water in the mesophyll tissue and in the spaces between the mesophyll cells. This causes evaporation to occur at those sites of evaporation, which then draws water up into the canopy. And the principle here is that water moves from areas of high water potential. Water potential is just you can think of it as the potential of water to move. So pure water has a water potential of 0. So hydrated soil, for example, has a water potential value of -0.1. As water is lost through the stomatal pores, the water potential declines, it becomes more negative. And this process is what draws water up through the plant. So this is really a remarkable biophysical process, and it's a passive process, at least in terms of the investment that the plant provides. Under conditions of moisture stress, so when the soil water potential declines, i.e. becomes more negative, it moves further away from 0. The plant water potential also declines as the plants are losing more and more water. Plants have evolved microscopic pores on the surfaces of the leaves to reduce this rate of water loss. So these are the stomata on the surfaces of the leaves. Stomata can close as the plant loses turgor, as their water potential declines. However, water loss continues even after stomatal closure occurs, because stomata are leaky, and there's also particular water loss from the surfaces of the leaves. And what this may result in is air being drawn into the xylem. You can think of it as a rubber band. If that water column stretches as the plant continues to lose moisture, eventually that rubber band can snap. And I've been fortunate to be involved in some research that's developing tools that allow us to detect when these embolism events, these air occlusion events occur. And I'll just demonstrate here with a video. So how we do it. So this is an image of the surface of a leaf. So we're looking at the light that's transmitted through the leaf. And if we collect multiple images of the same section of the leaf as the plant dries down, and we look at the image difference, we can detect the difference between xylem that is air-filled and xylem that is water-filled. So this is now a plant that's drying down, and so the water potential again is decreasing from 0 to more and more negative. And eventually, as this rubber band, as this water column is stretched, we should start to see some embolism events shown on this right panel here. And this is where gas is entering into the xylem of these plants and forming occlusions which block the transport of water through the plant. We can define a typical sequence of response of a typical plant during periods of water stress. As I mentioned, the plant's first response is to close their stomata. So a fully functional plant is happy and healthy. It's got open stomata, it's hydrated. As the plant dehydrates, it closes its stomata. Some plants are drought deciduous. They shed their leaves to avoid even further water loss. If the water stress continues, we start to observe embolism in the xylem vessels of these plants. This is a non-recoverable process. This is a damaging process. This is the start of lack of recovery following rehydration. If the drought event continues, the plants can lose function in the distal shoots. If there's some water left in the bowl, they can still recover if they are able to resprout, but eventually the plants succumb to complete hydraulic failure, complete plant dieback. So, okay, the first study that I'm going to relate to today is one that I started during my PhD and I've been revisiting upon returning to the mountain Fynbos. And it was a study where we used these principles of plant physiology to quantify the loss of function during dry years. And during my PhD, I worked at this beautiful site, Johannesburg, in the Riviersonrent mountain range in the Western Cape. We monitored the physiology of 3 species, this overstory Protea repens, the canopy species Erica monsoniana, very fine-leaved, shallow-rooted species, and then a graminoid Canomoeis congesta. And we collected detailed physiological measurements on these 3 species. Don't worry about every single point here. The idea that I'd like to relate here is the trajectory of these different species and how they differ. So what we see, potentially unsurprisingly, is that the shallow-rooted Ericas during the drier year, so season 1 was a dry year, season 2 was an average year. During the dry year, the water potential declines because it's a Mediterranean type system where we have long, hot, dry summers. I'm not sure if you can see my presenter views in a way, but here's a diagram of some of the Met variables. So the bottom panel here is rainfall. This was a particularly dry year and then an average year. And again, so the shallow-rooted Ericas decline in water potential. They become more dehydrated during the summer, potentially unsurprising. The deep-rooted Protea, shown here in white, is relatively consistent. Some decline towards the end of the summer. We think that that's a result of deep rooting in this particular species. The surprising result, however, is that the shallow-rooted Canomoeis also maintains water potential at relatively high values. Remembering that the higher the values, the more hydrated the plant. And in fact, the trajectory of water potential, even though in Erica and Protea it's fairly linear, in the Canomoeis, if you follow these black dots, there are points where the plant is somewhat dehydrated and then it hydrates and then it dehydrates towards the end of summer. And this is not following the typical rainfall trajectory that we recorded at the site. So the question is, what moisture sources are the restios able to utilize? And we think the answer is dew. Here's an example of a clear, you can see the clear sky in the background with all these droplets forming on the culms. And the architecture of the plant is such that any droplets that form on the surface of the culms is likely to run off into that dense fibrous mat of roots at the base of the plant. And in fact, we had a leaf wetness sensor out at the site, and we were able to demonstrate that dew is a relatively frequent occurrence at the site. And again, we think that this alternative moisture source is something that these restios are relying on. So if we return to our principles of physiology, so we quantified using that optical technique, we quantified xylem vulnerability curves shown as the solid sigmoidal curves for these 3 species. So one thing to note is that the species differ in their functional types, their functional traits. They differ in the xylem vulnerability to embolism. the erica is the most resistant, the Canamoes being the least resistant. And how we define resistance is based upon this one metric called the P50, the water potential at 50% loss, which is indicated by these dots in the middle of the curve. And this is a point of substantial change in function. It's a point of inflection on these sigmoidal curves, representing a period of a water potential associated with extensive damage to these plants. So we can combine the water potential data that we've collected from the field with the lab-based measures of these functional traits, the xylem vulnerability curves, and what we can what emerges from that is a prediction of loss of function experienced by these species simply as a result of hydraulic failure. And so I've tried to visualize the extent of the damage that we saw during this dry year. And so if this panel here on the right represents the total cumulative embolism in an erica in this case, and Canamoes in this case on the right. The middle panel indicates the damage associated with the drier year shown here in black. So this is the water potential of this dry year. And we can see that the erica and the Canamoes are both expected to have incurred some non-recoverable loss of function as a result of hydraulic failure. The protea, again, because it's deep-rooted, it seems to be able to avoid these water potentials associated with non-recoverable loss of function. So that's the prediction based on the traits and some of the physiological measurements. We developed and built our own miniature sap flow gauges that we use to monitor the water flow through the plant. And what we were able to show, again, comparing the dry year with the wetter year, is that during the dry year, the ericas lose function incredibly rapidly. You can compare the wet year response, where the plants remained relatively hydrated, to this decline in summer. So we know that these summer dry conditions are making these plants lose function. And then the question is, how much of this loss of function is non-recoverable? And what we used to determine that is we compared the recovery of gas exchange following a rehydrating rainfall event shown here in blue. This vertical dashed line is the actual rainfall event, and the blue is periods of relatively wet conditions. This is essentially the return to the hydrated state. And what we can do is we can compare the maximum values of a fully functional plant to the recovery based on these on a return to rehydrated conditions following a dry condition. Another thing to note here very briefly is that again, the restios show a much slower decline of function. So this is Canamoes, the restioid. The loss of function is a lot slower in this species compared to the erica. And again, we think that that's because of these alternative moisture sources, which are important in the context of thinking about global change. We really need to get a handle on some of those alternative moisture sources. And then the protea, which shows a very linear decline. Another thing to note very quickly is that the ericas seem to be responding to the summer rainfall events to an extent far greater than some of the other two species. So what does the data suggest? This is two panels showing the lack of recovery of gas exchange following this rehydrating rainfall event. This top panel is transpiration. That's the rate of water loss through the plant, and this bottom panel is hydraulic conductance. This is the ease with which water moves through the plant, and this is the process that the embolism prohibits. And as expected, the Canamois and the Erica are both suffering loss of function, loss of transport capacity through the xylem. That's predicted from the embolism, from the predicted embolism from the xylem vulnerability curves and the water potential. The surprising result here is that the proteas also seem to experience the lack of recovery. And we have, I won't go into the details on the explanations, but there are two possible explanations for why the proteas aren't recovering following the rehydrating events. One is the accumulation of phytohormones, where the plant essentially accumulates abscisic acid, which then prohibits the reopening of stomata following recovery. And the other is that we quantified the xylem vulnerability curves on stems rather than leaves. And what we observed was some damage to the tips of the leaves of some of these proteas, shown here in this inset. And the idea here is that there might be what's called segmentation between the leaves and the stems, where the leaves are more vulnerable to embolism than the stems. And the idea behind this is that the stems are the more valuable tissue to the plant. So they essentially sacrifice the leaves to protect the more important stems from which they can resprout and regenerate. But the important thing to note is that the physiology enables us this pathway to predict the recovery of function, which seems to be severe in the loss of function seems to be severe in these mountain Fynbos systems. The other thing to note briefly here is that the loss of recovery at the whole plant level seems to mirror the findings from another site on different species of whole plant mortality. So this is a study by Adam West and colleagues from the University of Cape Town at Silvermine, where they imposed an incredibly severe drought. So this is so our dry year was it was a natural dry year. This experiment here is was a severe drought where they removed all rainfall. So they essentially pushed these species to their limits. And what you see is that in the drought treatments, there's a high mortality of Erica individuals at this site. So there seems to be a direct link between the physiological function and the loss of the physiological function and the drought mortality outcomes from a severe drought experiment. So this suggests that this suggests two things. Firstly, that we can use physiological principles to predict these drought outcomes. And then secondly, because the results are in agreement, we can start to talk about functional type responses, where we talk about ericoid responses and restioid responses and proteoid responses. So the second case study that I'd like to briefly touch on is one where we are trying to use the physiology to infer and track changes to plant status through time. To do so, we sampled water potential. The screen is mostly dark because we sampled water potential in the dark mostly. We did a lot of measurements before the dawn. And what we were doing was we were following up on a historical data set of water potential that was collected 40 years ago by some Americans that came out, and they measured a whole bunch of species at a range of sites. And we were looking to detect whether there's been any noticeable change in the water status of these plants. And to give you an idea of some of the expected changes, here I've selected two sites that these researchers worked at. The orange arrows indicate the years in which the water potential was sampled by the Americans, and the sites are Jonkershoek, in the Southwest Cape, relatively high rainfall, over 1,000 mils mean annual rainfall, and then Algeria in the Cederberg, lower rainfall, around 700 mils. Some interesting long-term trends in rainfall from these sites are at Algeria, rainfall seems to be declining. There's a significant decline in rainfall, mean annual rainfall at the site, and this is mirrored by the rainfall, a decline in rainfall in the driest third of the year, which is December, January, February, March, the driest 4 months. And but at the wetter site in the southwest corner, there's been, excuse me, there's been no real noticeable change in mean annual rainfall, although a slight change in summer rainfall has occurred at this site. So we sampled years that were very similar at Jonkershoek in terms of mean annual rainfall, maybe slightly lower summer rainfall, which might affect the minimum water potential that I'll be reporting on. At Algeria, at the more northerly site, we sampled in a much drier year, and so the prediction here is that the plants should be more stressed at the Algeria site. So what do the results suggest? Firstly, this is the historical. This is just a small section of the small sample of the historical data set that many more species were sampled, but this is just a few of the species at these two sites, and I'll be presenting here the midday values. We sampled at pre-dawn and midday, and so this is the R sampling. The thing to take from this is that there's no obvious trend that emerges when you look at a species level. There's some at the drier site. There's some species that do show a decline in water potential with lower rainfall availability, but this trend is not consistent, and for the most part, the values are relatively close to the historical. At the wetter site, interestingly enough, some of our species were more hydrated than what was reported in the past, and the various physiological reasons for why this might be the case, which I won't go into now. The aspect that I want to present is when we scale up from, or when we summarize from the species level to a functional type level, we can see that some of the patterns that start to emerge. So what we detect is that at the drier site in the Cederberg, Algeria, the mid-shrubs in particular seem to be more dehydrated, and again, this is likely to be linked to a decline in summer rainfall, which we think they are reliant upon. There's some very strange results where the geophytes are more hydrated, and this possibly has to do with the plants shutting down earlier in response to greater aridity. The restios pre-dawn, they were more hydrated than what was reported in the past. So these are interesting trends that we think we can start to unpack from a physiological level. And again, part of the explanation is that we start to see very specific responses of these different functional types as a result of them being reliant on different moisture sources. So the mid-shrubs, the ericoids, they tend to be relying upon summer rainfall. So when summer rainfall declines, we see a decline in water potential. Again, in line with the predictions that the ericoid functional type is likely to be most impacted by global change. The restioids are an interesting example of where we think that they're relying upon dew moisture, maybe cloud moisture, mist moisture, fog moisture, and to understand why these plants are more hydrated in conditions when summer rainfall has decreased, it might be necessary to understand changes in alternative moisture sources. And I think this is an avenue of research that really needs to be addressed, is how are some of these other functional types, like restios, which are important in Cape Mountain Fynbos regions, how are they likely to respond to changes in temperature, which then influence dew availability and alternative moisture availability. Another intriguing element there is that this might not be restricted to the restios. John Rourke, in his magnificent Protea book, commented on Protea pruinosa, which occurs at the very high altitude band in the Swartberg, very arid sites. He commented on the large size of these leaves and how unusual or how surprising they are for such an arid site in the Swartberg. And he speculated that these leaves, which are they're shaped in such a way that they might be able to trap dew and allow dew to run off into the roots. This may also be related to why these plants are prostrate. They're very low growing. Maybe they're trying to connect maybe this is to connect the moisture sources collected on the leaves to the roots. So there are all sorts of interesting avenues that I think can be explored in these mountain Fynbos regions. And this is I think the last reason why I think investigating plant physiology is such an important and such an interesting discipline. It allows you to examine the evolution of plant form and function in a way that's meaningful to try and understand how these plants are likely to be responding to global change events. So with that, I'll wrap up. I think I'm probably right on the edge of time. So I'd just like to, again, maybe this is preaching to the choir for a SAEON seminar, but I'd just like to highlight the importance of monitoring the local environment, monitoring local met variables, in addition to rainfall, in addition to temperature and relative humidity, things like fog availability, things like dew availability. These are potentially incredibly important drivers of plant function in these natural vegetation areas. And when we couple monitoring the local environment with monitoring plant performance, we can start to understand the potential impacts of abiotic stress and global change on these natural systems. To do so, it's important to invest in research tools, the development of research tools that allow us to quantify these things like SAMPLO technology, things like STEM psychrometers, which allow us to monitor plant water status. These are incredibly important tools that allow us a window into understanding how these plants are functioning. One thing that's become apparent for me, at least, is that biodiversity is often associated with incredible functional diversity. In the Cape, we have thousands of species. There are at least 9,000 species in 90,000 square kilometers. It's an incredibly biodiverse region, and this goes for other areas of Southern Africa as well. And this biodiversity, as we've seen with variation in xylem vulnerability curves, variation in rooting depth, variation in leaf size, this biodiversity is linked with functional diversity. And this means that different species are likely to be responding very differently. And we need to get a handle on how on how some of these species are responding. Fortunately, I think it is possible to use physiological principles to predict how these species are likely to be impacted. The way to do that appears to be to use a functional type approach, where we group species into functional types based on their functional traits. And for this, it's important to be able to quantify those functional traits. With that, I'd like to acknowledge some of the collaborators that I've been lucky enough to work with. Adam West has been a long-term colleague, working from my PhD days to the present, and a few others that have hosted my research, Tim Broderick at the University of Tasmania, and then the Royal Society. I was very lucky, very fortunate to have received a FLAIR fellowship, which allows me to conduct the research that I am. The African Academy of Sciences is a partner on these FLAIR fellowships. The Academy provides support for the fellows, and they've been incredibly instrumental in the research. I'd like to thank SAEON for being the host of my research. It's incredible. It's an incredible organization that has a wonderful mandate of monitoring our very, very special environment. And then lastly, organizations like Cape Nature, which give us the permits which allow us to conduct the research in their reserves. And then lastly, just to thank everyone for listening to the presentation.
Thanks, Rob. There's a really, really interesting talk, and really, I think you're really changing the way that, or deepening our understanding of how Fynbos functions. And from SAEON's perspective, it's really great to gain this level of understanding. We do a lot of ecosystem level monitoring. We've got a lot of infrastructure in place to observe and monitor the ecosystem scale signals, like fluxes of water and carbon. But those signals are really the integrated product of all these individual plants and species. And if we don't understand what the individual species and the individual plants are doing, we actually don't understand those integrated signals that we receive from our larger scale equipment. So I think this is really critical work to build the science around a lot of the science and observation that SAEON's doing. So thanks. I have some questions myself, of course, but I'm going to open the floor to anyone who has a question. I think the best way to do that is to raise your hand. If you do that by clicking, there's a little reaction button and raise hand. I will try go through those people who've got their hands raised. I can see that Tony Swimmer has a question, so go ahead, Tony.
Hi, thanks, Glenn. Yeah, thanks, Rob, for an interesting talk. So if future droughts wipe out the aerocoids, as your work suggests, then I'm wondering about what will replace them. And the first thing that comes to mind is grass species, because we have a lot of drought-tolerant grasses that could handle that those sort of water potentials. So I'm just wondering, is there any of this type of physiological work that's been done on grasses that could potentially replace those aerocoids? Or alternatively, is there some other functional group within the Fynbos flora that you see moving into that niche?
Yeah, thanks, Tony. That's a great question. And I think the best way to answer that is to say so grasses are not terribly prevalent in the Western Cape, in the Fynbos. I think there are a handful of grass species, of indigenous grass species in this region. Now, I suspect that it's unlikely that the grasses will be the functional type, that the gramineaults will be the functional type that take over. I think the likeliest replacement group will be plants from the succulent Karoo, some of the succulent species which can occupy a more ephemeral water surface, soil water layer. So I think the succulent karoo might shift southwards essentially. That would be my intuition. And I think there are a few studies which are looking at the interface between Fynbos and succulent karoo. I know Adam West from the University of Cape Town has a study at a reserve right on that interface. And those results are currently being published. The grasses do come in, I should say, in places like Table Mountain, where the fire regime has shifted. So where there's more frequent burns, the grassy, there are a few graminoids and they do tend to proliferate. So it's not beyond the realms of possibility that the graminoids might become an important player in this region. To answer your question on whether there's been much physiology done on the grasses, yeah, not in a Southern African system. Grasses graminoids are an incredibly important part of other systems. Grasslands, obviously, savannas, but also a lot of the agricultural crops are graminoids, so oats and barley and wheat and all these kinds of annual crops. So there has been a lot of work done, but more in the kind of agricultural sector. And it is important to say, I guess, at this point that there are examples of where these physiological tools have been used on grasses. So I can think of a study where the optical technique has been applied to wheat and to try and examine the drought tolerance of wheat.
Thanks, Tony. Rob, is there? I don't see any other hands raised. Is there someone else who has a burning question? We've got 5 minutes or so for questions. If there's no one going to raise their hand right now, there's a question that I have. Rob, when you were showing your results of the sort of loss of function that occurs after a drought, the drought that you were showing seemed like it was essentially the normal summer drought, the seasonal summer drought that we have in the Cape, and there was a certain level of, even for the rest year, which seemed to recover well, as far as I remember, there was a certain level of non-recover. Nothing recovered to 100%, and the Ericas and the rest years obviously didn't recover very well at all. Of course, that drought is regular. Every year we have that drought, that summer drought. So does that mean, how do we interpret that? As a plant basically losing every year a certain percent of its function, is it dying back? Is that kind of the annual setback that it suffers and it has to come back from that?
Yes, so I think that's right. I think this is part of the reason of why plants grow, right? Why they need to produce new leaves every year. In addition to drought damage and abiotic stress, there are also biotic agents. So things like fungal pathogens, things like insects, and maybe not in the Cape system so much, but in some of the savannas, the herbivores. So there is a natural loss of function. And if I can go back to those results, let me see am I still sharing my screen? Am I still sharing this?
Yeah.
Okay. So this was the idea behind trying to quantify loss of function in what we consider to be a normal year. And so even in a normal year, you do have fairly substantial lack of recovery. So there's up to 20% lack of recovery in these species, and this is in a good year. And So the idea is that I guess life is hard for a plant, right? In a Mediterranean type system. So you some of the conduits don't develop properly, and they experience embolism in the absence of abiotic, in the absence of drought. You have insect damage, which reduces the leaf area, reduces the function of these plants, and yeah. So even in a normal year, in a reasonable year, in an average year, you still see loss of function. In these dry years, we were trying to highlight two ways in which abiotic stress is likely to reduce function. The one is within the season. Within the season, these plants have low function. So this is best shown with the erica, where in fact, I'll bring that slide up. So if you compare this function here with the average year, these values are much lower. And to understand why this is important, there's a relationship between sap flow, which is equivalent to transpiration, which is related to stomatal function, and assimilation. So what I didn't mention was that in addition to water being lost from the surfaces of the leaves, the reason why plants have stomata in the first place is that they need to take up carbon dioxide. So the lower the function within the season, the lower the carbon dioxide uptake. So this in itself, even if this is 100% recoverable, this is a loss of function for this species in this case, and for this entire community if you scale up. So and that's in the absence of non-recoverable loss of function. So even in this, for example, even in this wet year, towards the end of summer, the stomata start to close, which then affects the ability of these plants to take up carbon dioxide. So if we translate this to an agricultural setting, this has importance for things like irrigation. So in the Cape, farmers might wish to irrigate at this point to increase the carbon capture ability of these plants. Does that answer your question?
Yeah, no, it does. Thanks. I think it's really interesting to kind of think about it in terms of if you have control over that irrigation, when is the best time to do it? Is it before the plant loses? Can you prevent the loss of function, or do you wait until it's at its bottom and then water? So I think there's definitely some, kind of agricultural applications of this as well, when there's some sort of human intervention in this process.
Yeah, and it varies by species, right? So grapevines, for example, the more moisture stress grapevines experience, the better the sugar production into the grapes, and the better the quality of the wine. So, exactly how that exactly what you do with this information depends on the ultimate goal. I'm not sure how we mitigate the effects of abiotic stress and water stress in these natural systems. I guess this is a plug for being responsible citizens and trying to mitigate our carbon production, to try and mitigate the extent of change in these systems. But yeah, in an agricultural setting, it might be more, we might be more able to use this information to drive the desired response.
Thanks, Rob. I'm gonna close it there because it's 11 o'clock now. But just for everyone here, I just want to advertise next month's seminar, which is on the 2nd of July. And the speakers are going to be Nicole Duplessis and Erica Brown from Agagasini. And they're going to be talking about ecosystem accounting and ocean accounts, the what, why, who, and how. So that'll be our next seminar. But yeah, thanks on behalf of everyone, Rob. And yeah, we'll see you all next month. Cheers, everyone.